Sorry.
You want to introduce yourself? Our next speaker is Dr. Kavita Rangan. She received her PhD at Rockefeller University in the lab of microbial pathogenesis and chemical biology with Howard Hang. She's now a postdoc at the Rex Peterson lab at UC San Diego, and the title of her talk is RNA recoding in cephalopods, tailors microtubule motor protein function. The stage is yours.
Sorry. Oh, no. Oh, cool. Okay. Yeah, because it did it with presenter viewing. All right, sweet. Okay. Yeah, I guess I just wanted to tell you about some of the stuff I've been doing looking at RNA editing in cephalopods and how these animals use recoding to basically tailor the function of their microtubule motor proteins in different environments. And then at the end, I thought I'd just touch on some ideas I have for the future. So I thought I'd start by just saying, like, super broadly that I'm interested in how organisms acclimate and adapt in response to changing environments. And I like thinking about RNA editing because to me it really illustrates kind of the interdependence between the genetic and the environmental in the context of an organism. So I thought I'd start with, the so-called central dogma of molecular biology and, one textbook explanation for why we have these intermediary steps between DNA and protein is that they, of course, provide a space for all kinds of regulation. And one type of regulation that occurs across all domains of life is RNA editing. And I think RNA editing is interesting from an evolutionary perspective because I think it really muddles the lines between these different types and levels of information in an interesting way. And so, for example, RNA editing offers like a potential mechanism by which an environmental stimuli can lead to the diversification of protein output from any given RNA. And one of the most direct ways that this can happen is through a process called recoding. So in animals, one major type of RNA editing is mediated by these enzymes called ADARs. And so they basically convert adenosine to inosine in double-stranded RNA. And they serve really diverse functions in the cell. And one of those functions is the editing of mRNAs. And so it turns out that inosine is actually read by the translational machinery as a guanosine. And so when editing happens in coding sequences, this can result in non-synonymous codon changes. And so that's what's called recoding. And so this is cool because it means that editing can basically generate proteins with different amino acid compositions and different functions. And so in humans and some common model animals, recoding through RNA editing is actually relatively rare. But what's interesting is that recoding occurs really extensively in the soft-bodied cephalopods. And so that's in squid, octopus, and in cuttlefish. So in these animals, basically 60% or more of their transcripts are recoded through RNA editing. And so, this really extraordinary level of RNA editing, kind of RNA recoding, makes us wonder, like, what's the function of this process in these animals? And so recoding sites bear sort of signatures of positive selection. And so one idea has been that recoding might be a mechanism for these animals to kind of dynamically diversify protein function in different contexts. And so if you think about it, this process is really generating massive amounts of protein variation, but it offers kind of a potential way for these animals to kind of flexibly respond to their environment. And so we can ask, is this actually true? Is recoding being used to regulate protein function in different environmental conditions? So if we take a step back and just look at, for example, what Go terms are enriched amongst edited proteins in the squid, we see that terms related to transport and localization are overrepresented. And this might just reflect that editing is actually important for the regulation of these processes. And it turns out that microtubule-based transport proteins are really heavily recoded in these animals. So here I'm just showing the number of recoding sites that were identified from these 4 species of cephalopod across the kinesin 1 transport complex and the cytoplasmic dynein transport complex. And all I really want to point out here is that there are just hundreds of recoding sites across the proteins that make up these complexes. And so basically I decided to come to the Rangan lab because the Rangan lab specializes in microtubule-based transport mechanisms. And I think microtubule motor proteins are a really good model to explore the effects of RNA recoding on protein function. So, dynein and kinesin are these sort of multi-subunit protein complexes that actively transport really diverse cargoes in the cell. But what's nice is that they're really robust in quantitative assays for their function. And then there's also just a real wealth of structural and mechanistic insight into their function. And so one of the ways that we can look at motor protein function is using single molecule motility assays. So in these assays, polymerized microtubules are adhered to a cover slip, and then fluorescently labeled motors can be flowed in. And then motility can be directly imaged through turf microscopy. So here I'm just showing GFP-labeled kinesin walking along a microtubule track. And so from a video like this, we can make chymographs where you can see the movement of the fluorescent motor moving along the microtubule over time. Then we can analyze these chymographs for different parameters of motility. Okay, so the question is, does recoding actually diversify motor protein function in these animals? And the aspect that I'll sort of touch on for this talk is how recoding might interface with the environment. So for example, is recoding sensitive to the environment? And then are differentially recoded protein variants actually functionally distinct? So for this work, I really focused on recoding of kinesin 1 in the squid. And so, our first question could be, what kinesin variants are actually being generated in these animals? So from RNA sequencing, we know that there are 14 recoding sites across 10 amino acids just in the kinesin motor domain alone. But we don't really know how these sites exist in combination with each other. So what variants are being made and how that might change in response to the environment. So one environmental factor that just comes to mind right away as being really important for these animals is temperature. So the California market squid, its story-toothed opalescens, it basically migrates all along the Pacific coast from Baja to Alaska, and it spawns right off the coast of San Diego, which provides us access to animals. And temperature is just a really important factor for these animals. And so, not only do they do these long migrations, but they also do these daily depth migrations. And this data here is showing squid sightings, basically at various depths. And then the blue line is showing temperature. And so you can find these animals in quite cold waters down to 5 to 6 degrees C. And so we can ask, is editing in squid actually sensitive to ocean temperature? And then could temperature-specific recoding actually alter protein function at different temperatures? And so temperature has been shown to sort of globally affect RNA editing in other animals. So for example, in flies and in even hibernating squirrels. But what this is doing for these animals isn't very clear. And kinesin motility is obviously temperature dependent. And so here I'm just showing human kinesin, and you can see that human kinesin has shorter run distances and slower velocities in the cold. But humans' body temperature doesn't really change that much. So it's kind of interesting to think about how squid kinesin might behave at the temperatures that these animals experience, and whether recoding might be important for modulating that function. So in order to kind of test the effects of ocean temperature on recoding of kinesin in squid, I did experiments with hatchlings. And so basically I just took hatchlings and put them in tanks of different temperatures for 1 day, and then just extracted RNA and then looked to see what the editing looked like. And so here you can see that across most of the sites, recoding sites across the kinesin motor domain, editing actually increases as temperature decreases. And I think what's kind of cool is that we see these changes quite rapidly, so in less than 24 hours. And so, the next thing that I was curious about is, how do these recoding sites actually exist in combination along individual transcripts? And so to look at this, I basically just cloned out from cDNA and then sequenced a bunch of clones from animals that were exposed to either 6 degrees C or 20 degrees C water. And so here, to just summarize, you can see that there's a much higher proportion of completely unedited transcripts from animals that were exposed to warmer water, and then more heavily recoded transcripts in animals that were exposed to cold water. And here, just taking a look at the sheer number of unique sequences, we can see that recoding is really generating just many, many temperature-specific kinesin variants. And so, one thing that I was curious about is whether editing at each site is occurring independently or whether editing at distant sites is actually correlated. And so here, I'm showing a pairwise correlation analysis where on each axis is a recoded nucleotide position, and then a colored box would indicate a significant correlation. And so what you can see right away is that there are significant correlations between recoding sites, some of which are distant, and that there's actually temperature-specific correlations between recoding sites. And so this kind of suggests that there are, potentially many mechanisms regulating combinatorial editing in these animals. Right, so I wanted to basically test the motility of different kinesin variants and sort of see if variants that were generated in the cold, how they would behave in the cold. And so to do this, I basically did single molecule assays with a temperature-controlled stage to be able to look at motility at different temperatures. And so here I'm showing unedited squid kinesin. And you can see that much like human kinesin, it has slower velocities and shorter run distances in the cold. So next I wanted to look at basically 3 different kinesin variants that were identified exclusively in animals exposed to cold water. So here I'm just showing unedited kinesin again. So like I just showed you, it has slower velocities, lower run distances. And then here I'm showing landing rate, which is the number of events per distance per time. And you can see that in the cold, the landing rate is much lower. And so then next I looked at these 3 different kinesin variants that were found from animals in the cold. And you can see that 2 of these variants displayed slightly reduced velocities, but significantly increased run distance. And that all 3 of them significantly increased landing rates in compared to unedited kinesin. And so I think, this kind of shows that recoding in response to cold water is actually generating kinesin variants that are able to bind the track better and travel further in the cold. And so then next I basically wanted to break this down a bit. And so I looked at 3 different cold preferred recoding site substitutions and looked at their activity in human kinesin. And what I saw is that each of these 3 individual substitutions actually increased run distance and increased landing rates of human kinesin. And so I think this goes to show that, these recoding sites can actually inform the characterization of even human kinesin. And I think this kind of speaks to an idea that might be interesting, which is that, if recoding site substitutions represent functional variants in these animals, can we use these sites as a guide to kind of interrogate protein function? So I think, it can be appreciated that it's difficult to kind of predict how any given amino acid substitution might alter the function of a protein. But, if we look at recoding sites, we see that many of these are in regions that are sort of uncharacterized. And they might provide kind of a lens for us to focus on, functional substitutions. And so to try and approach this idea, I've looked at the intersection between sites that are recoded in cephalopods, but that are broadly conserved. And I've used kind of kinesin and dynein as test cases. So, for example, here's just the structure of cytoplasmic dynein 1 heavy chain from human. And you can see in black are highlighted all of the residues that are recoded in cephalopods, but that are conserved between cephalopods and human. And so, we can ask, how do recoding site substitutions at these sites actually affect motor protein function? And so I've taken this approach with and looked at a bunch of different sites in human kinesin and yeast dynein. And despite how much we know about these proteins already, a lot of these, for most of these substitutions, we wouldn't necessarily be able to predict their effects on motor protein function. And all I kind of want to point out here is that, the surprising thing is that, the majority of the sites that we looked at actually don't abolish processivity. So unlike disease-associated mutations, they don't, really abolish processivity, but rather seem to kind of fine-tune other aspects of motor protein function, for example, velocity or run distance. And I think it kind of speaks to the idea that recoding sites might be an interesting resource, beyond structure and homology alone, to kind of ask, how are cephalopods diversifying their protein function through recoding, and can we learn something from that? And so, to summarize, I just wanted to say, recoding is kind of a dynamic mechanism in these animals for regulating protein function in potentially different environments. And, we might be able to look to recoding sites to kind of highlight residues of interest in non-cephalopod proteins as just an interesting auxiliary tool. And so, going back to that schematic that I showed in the beginning, I just showed you, that changes in water temperature can lead to the sort of the alternative recoding and diversification of motor protein function. And this might be sort of an important mechanism for regulating transport function in these animals. And I think this all kind of supports the idea that editing is a mechanism that generates phenotypic plasticity in these animals. But what's cool is that, because recoding is just so prevalent in these animals, not only are motor proteins recoded, but proteins involved in basically every cellular process are. And so for my future work, I've become really interested in this idea of that I'm calling, this kind of strange loop, where basically the machinery involved in DNA level inheritance is recoded. And so, from basically a developmental and an evolutionary perspective, I think this is really intriguing because it suggests a mechanism by which environmental stimuli like temperature can directly impact DNA level inheritance through the dynamic recoding of proteins involved in DNA replication and repair. And so thus it might be important, for example, in adaptation. And so to kind of, as a first iteration of this idea, I'm interested in looking at how recoding alters the function of DNA polymerases. So for example, looking at DNA polymerase fidelity and processivity. And I think this is really exciting because it might kind of show us new ways that we can actually tinker with polymerase function in the context of temperature. And then sort of taking a more organismal approach, I'm also interested in looking at how recoding alters things like mutation rates and mutation bias, and thus might affect fitness or adaptation. And then as kind of like a final thought, RNA editing happens extensively in the gametes of diverse species. So, for example, even in filamentous fungi. And so I'm also interested in kind of exploring how editing might affect inheritance or development through inherited RNAs. And so, overall, my interest is to basically look at how does editing and recoding contribute to phenotypic plasticity and adaptation. And as kind of like a general approach, I'm going to look at basically analyze the editomes from diverse species and then functionally assess, recoding sites and recoded variants, starting with polymerases. And then kind of to further look into the role of editing and adaptation, I plan to do basically look at mutation accumulation and fitness in the tractable model fungus Neurospora crassa. So with that, I'd like to say thanks to everyone in the lab and then a bunch of my friends and collaborators for their support and help. And then thanks also to Leading Edge because this has been one of the best conferences I've been to. Anyways, thanks.
Thank you so much for this. Oh my God. Okay, what just happened?
I think you lost your stuff.
I lost it. It's fine. Thank you so much for this wonderful talk. It's so cool. I saw we have a question.
Yes.
Amazing talk. Thank you for sharing that. So you pointed out, I loved the bit that you have at the end, and I'm sure there are a million cool things to talk about there, but I love the trafficking as well. You showed that there are cargo adapters that get edited. Do you think there's any possibility that those edits, instead of affecting enzyme function, are affecting what cargo is getting coupled? That maybe there are some like endosomal targets that are more useful in cold.
That's totally possible. So so basically because we have so much just structural info of the entire, for example, dynein transport complex, basically building like a big map of all of the edit sites that are across cephalopods, but also the ones that are sort of dynamically changing with temperature, and try to see, like, are there any themes, for example, across cargo adapters, or whether it's just like but I will say with kinesin, there's a huge number, like way more edits are actually happening in the tail, so in the cargo binding region, than are happening. And similarly with dynein, there's just a ton of edits, and basically to your point, but we first started with just like this idea.
That's so cool. Thank you.
Yeah, that was awesome. I have a lot of questions. I'll try to limit myself to one. So you were talking about how these squid travel, like, do they live, does one squid travel up the length of the coast? Or does it have different
I guess
lifetimes?
I was a little bit, like,
I don't care the
implying,
answer. I'm
well,
just
yeah, no,
curious.
so they basically, so they'll spawn in multiple locations. Yeah.
Oh,
So they
okay.
basically travel and it's like a spawning migration.
Got it.
Yeah.
Okay,
But
so
I
like,
actually don't know whether like one squid will make a journey like across. I think so.
so like one generation could adapt to a certain temperature, and then the next generation could like.
So I think it's a lot faster, like in
Okay.
terms of this. I think this is way more about just like
Right, because they also
immediate
dive.
acclimation. And I also should say that like, we don't really, so because they do these kind of daily depth migrations. And I think what's more important than maybe invoking the idea that the actual pool is changing dramatically, I think it's just about establishing a diverse pool. So for example, the phenotypes that we saw, which are just, increasing binding to microtubules, you don't, even if you have a rare variant there, it will be sort of like functionally enriched in vivo in theory.
Mm-hmm.
So if it's just present in the pool of kinesins, for example, it would be, does that make sense? So I don't necessarily think you have to invoke that like there's turnover, complete turnover on the timescales that they're experiencing, but rather that
Like
this
you'll have
change
a
in
population
temperature
of
is like
motors that's more
exactly,
diverse.
yeah.
Do
Just
you
having
see
like a
any-
diverse selection.
Have you like pushed the temperature up, like?
To make it warm and
Yeah.
see. Yeah. So actually, the two temperatures that I looked at, 6 and 20. So 12 is kind of their preferred temperature.
Okay, so 20 is.
But 6 and 20 are kind of just super extremes on either end. Yeah.
Cool. Thanks.
Okay, we have a little bit time for maybe one or two questions. Don't fight.
Thank you. That was really fascinating. This is just a complete curiosity question because I don't know anything about this field. How much is known about RNA recoding during camouflage, which is really critical for all these animals?
Oh, yeah. So they, in terms of like how much does RNA editing contribute to camouflage, I don't think anything is known. But I would say that these animals are doing it in all of their tissues all the time. And so from that perspective, whether or not it's actually, whether it's like noise that's being buffered or whether it's like having some sort of specific functions is totally,
Okay. Yeah, I also have a curiosity question. So you showed like there are all these different species of squid. And so I was just curious whether they have different like ranges in where they live and what temperatures they're exposed to and whether there's differences in what's edited.
Yeah. So, like for example, so I guess previous work has shown that there was kind of this idea about temperature potentially affecting recoding because if you looked from 2012, Josh Rosenthal's lab published this data where they looked at Antarctic octopus versus tropical octopus. And so those animals basically exist exclusively in these different temperature regimes. And then they saw that there was like more recoding in the Antarctic octopus, it's whatever. And so, but then whether or not like in an individual animal you could actually get this dynamic recoding was kind of still an open question. But yeah, so how much of it, there's this kind of interesting evolutionary question about whether some of these sites are like hardwired in the sense that like they always fold that way and then you get editing, or whether they're like dynamically folding and getting different editing. Yeah.
Awesome. Sorry, no time for more questions. I'm sorry. Let's join me in thanking Kavi again.